Good day. Our group will be presenting the paper of Haas et al. In 2017, which is the comparative study of impaction and sedimentation in an aerosol chamber using defined fungal spore and bacterial concentrations. The problem of this study focuses on whether impaction is significantly more efficient than sedimentation for bacterial and fungal spore detection under standardized conditions. This study has two objectives: to identify standard methods of measurements for fungal spores and bacterial concentration in an aerosol chamber, and to analyze the correlation between the impaction and sedimentation under standardized conditions.
For our methodology, it will be divided into 5 parts, namely, description of the aerosol chamber, sampling methods, preliminary experiments, standardized test series with impaction and sedimentation methods, and statistical evaluations. For the description of the aerosol chamber that was used was an ISO PRO under pressure isolator. It consists of an aerosol chamber and a transfer chamber with a double door system, shown in 5, which serves to load and unload growth media, and the air sampler mass-100 NT. The isolator is equipped with protective gloves, shown in 2. It is also equipped with protective inlet and outlet filters, shown in 9 and 4, respectively. A ventilation system on the right-hand inside wall has a high-efficiency particulate air filter or HEPA filter classification H13. This sucks the air out of the chamber again. In addition, a constant maximum negative pressure of 300 pascals is generated, monitored by our pressure indicator shown in 3. There is also an axial ventilator on the ceiling of the aerosol chamber, shown in 8, and in the upper front area, there is a neon tube lighting the workspace, shown in 1. Both ventilator and fan can be switched on individually. The aerosol chamber is connected to the aerosol generator by means of a tube system. With the aerosol generator, shown in 7, solid particle aerosols can be generated from suspensions. Lastly, the flow rate of the aerosol generator is 3 to 10 liters per minute. Now for the sampling methods. Active sampling was carried out by a one-stage microbial air sampler, mass-100 NT, operating at a flow rate of 100 liters per minute. The cutoff size of this device is adapted to collect airborne microorganisms with the aerodynamic diameter of more than or equal to 2 micrometers. A disinfecting agent was used for cleaning the device after each sampling, and passive sampling, on the other hand, using the sedimentation method, also called open dish method. This is where the Petri dishes were placed centrally on the bottom of the aerosol chamber. Following a defined aerosol flow time, the plates were opened for the microorganisms to settle. For measurements and evaluations of the fungal spores, malt extract agar, or MEA, and dichloroglycerol agar, DG18, were incubated at 37 degrees Celsius for 24 to 48 hours. Testing of the bacterial strains was carried out using casein peptone soybean flour peptone agar, or CASO, and cholestyne nalidixic acid agar with 5% sheep blood, or CNA, at 37 degrees Celsius each for 24 hours. The relative humidity and air temperature were measured with the infrared thermohygrometer Testo 445 while conducting the test series in the chamber.
The preliminary experiment tested the following suitable measuring parameters presented on the screen. Fungal species Aspergillus niger and the bacterial strain Staphylococcus aureus were selected as test strains, as these microorganisms are particularly important for the health of humans and can easily be detected in aerosols. For the production of Aspergillus niger stock solution, a pure culture was initially cultured on malt extract agar and incubated at 37 degrees Celsius. Following an incubation period of 24 to 48 hours, a randomly selected portion of spores was suspended in 100 milliliters of NaCl plus clean 80 and filtered through a sterile gauze bandage. For Staphylococcus aureus, the cryo pellets loaded with bacterial test strains as aureus were placed in 100 ml sterile distilled water and subsequently shaken for 30 minutes using the SKI4 shaking incubator. For the determination of concentrations of Aspergillus niger, 100 microliter of the stock solutions were collected and dilutions of 10 to the negative 1 to 10 to the negative 6 were prepared. From each of these solutions, 100 microliters were plated in duplicate on malt plates and incubated at 37 degrees Celsius. In order to be able to determine the concentration of the bacterial suspensions, the samples were also plated in duplicate in different dilutions onto the culture media CASO and CNA and incubated at 37 degrees Celsius. Following the incubation period, the colony-forming units, or CFU, were counted, and the fungal, spore, and bacterial concentrations were determined in CFU per ml respectively. To find a suitable standardized procedure in active and passive sampling, a series of preliminary experiments was conducted. First, the conditions for the different measurements with the MAS 100MT and the sedimentation method inside the aerosol chamber were changed by alternately operating and not operating the outlet filter system and or HL ventilator. Afterwards, the obtained results were compared. Figure 2 shows a diagram of the air circulation inside the aerosol chamber. The active and passive measurements of fungal spores and bacteria were conducted prior to the aerosol inflow and after aerosol inflow in intervals. Frequency and time intervals of the active and passive airborne microorganism measurements were also modified after aerosol inflow. For the preliminary tests, the suspensions with defined fungal spore and bacterial concentrations, which were in the optimal countable range, were used for the standardized test series. 1 hour of aerosol flow was needed to fill an aerosol chamber with a volume of 0.65 cubic meters. The stability of fungal and bacterial suspensions were monitored in each measuring series at the beginning, after 1 hour, and after 2 hours by plating 1 microliter and 10 microliters onto the growth media in duplicate and incubating for 24 hours at 37 degrees Celsius prior to counting.
A total of 30 measurements of both actively and passively collected airborne microorganisms were done for standardized test series with impaction and sedimentation methods. The concentrations of the suspensions most suitable for the measurements were between 102 CFU per milliliter and 104 CFU per milliliter for the fungal spores, and between 103 CFU per milliliters and 105 CFU per milliliters for bacteria. From each duplicate determination on the culture media used, the mean value was calculated for the determination of CFU per milliliter. In the standardized test series, both measuring methods were conducted without operating either outlet filter system or HL ventilator. First step before measurements is the first stability monitoring of the suspension. Here, 10 minutes rinse with distilled water of aerosol chamber is done combined with the outlet filter system for suctioning off the air and disinfection to be sure that it was free of contaminations. With this, you have the filling of 100 ml of the standard suspension in the designated vessel of the aerosol generator. This required defined fungal spore and bacterial concentrations, which were monitored through stability controls. Next, the aerosol inflow was started exactly 1 hour prior to the measurements in order to fill the chamber. However, before the start of the aerosol flow, growth media and the air sampler were brought into the chamber by way of the transfer chamber and positioned in the center. Next will be aerosol flow of 1 hour, then a second stability monitoring was carried out, after which the active aerosol measurements were started. All measurements were carried out in duplicate. For the active measurements of airborne microorganisms, the air sampler mass-100 NG with a sampling volume of 50 liters per 30 seconds for Aspergillus niger and 100 liters per minute for Staph aureus was selected in the preliminary experiment. Following the removal of the air sampler and the growth media, the aerosol chamber was again cleaned as I mentioned a while ago, followed by another renewed aerosol flow for 1 hour. In the course of the subsequent passive measurements, 4 MEA and 4 DG18 plates were opened and exposed to Aspergillus niger for 1 hour. 2 Petri dishes of Caso and 2 of CNA were exposed to Staph aureus for 1 hour and for 2 hours, respectively. Immediately after 1 hour flow, a third iteration of stability monitoring of the test suspension was done. And then, culture media were then incubated, and lastly, CFU per plate and then CFU per ml were counted for sedimentation and test suspension, respectively.
Microsoft Excel was used to determine the linear relationship, correlation coefficient, and the proportion, as well as microbial recovery between impaction and sedimentation. For the calculation of the ratio between active and passive measuring methods, the results are the CFU per plate of the impaction were divided by those of the sedimentation method for each measurement of the 30 test series. The average was calculated and evaluated separately for the 2 growth media. To avoid the bias in starting concentration of microorganisms and the repeated measurements of active and passive methods, the researchers carried out statistical corrections.
The study found that for the detection of Aspergillus niger spores, active impaction is more efficient compared to the passive sedimentation, whereas the optimal measurement methods for Staphylococcus aureus depends on a number of factors, including time, airflow, distribution of the microorganisms in the chamber, agar media, and position of plates inside the aerosol chamber. The findings of the study may help improve the MEM, or the microbial environmental monitoring, in both industrial and clinical settings. This is important to keep the risk of biological contamination to a minimum. Here's the study by Haas et al., and here's the link to that article. Thank you for listening.